Hi there. I would like to thank organizers for inviting me to talk about an exciting project we are currently working on. In this talk, first, I will very briefly make a case for an urgent need in accelerating the current probe discovery pace. I'll then introduce an original concept that I hope is a way to make drug or probe discovery more time and cost efficient. And next, I'll show how this FRASE concept was successfully applied in a proof of concept study. And finally, we'll discuss the ongoing work and future plans. It is not an easy task to find out how many drugs and probes there are for how many targets. Over the last 15 years, several studies have shed light on those numbers. Apparently, there are up to 10,000 targetable proteins that are waiting to be investigated. And only about 400 of them have known ligands. Also, by doing simple math, one can estimate the rate of probe and drug discovery to approximately 50 unique probes for 30 targets per year. At this rate, finding a probe for every target would take many decades. Hence, new breakthrough technologies are needed to significantly increase the discovery rate. In the next few slides, I'm going to present an original concept that I believe is a step forward toward more efficiently discovery technologies. We call this concept FRASE, that stands for Fragments in the Structural Environment. In the picture, you can see an example of a FRASE rendered in thick sticks. A valid FRASE includes a chemically sound fragment of a protein-bound ligand, as well as pieces of the protein that are within a certain distance from this ligand fragment. We can also qualify a FRASE good if the ligand was extracted from a highly potent against, is highly potent against the respective protein. We use the FRASE concept to develop data-driven structure-based strategy to assemble potent protein binders directly in the protein's binding pocket. The only input data one needs to apply this strategy to a novel protein target is its 3D structure. The structure can be either experimentally determined or homology modeled. No assumptions needed regarding the putative ligand binding site. FRASE can be considered as a key that joins together and exploits multiple pieces of highly heterogeneous data. In FRASE-based design, ligand fragments for a target of interest are distilled from the FRASE database that makes use of tens of thousands of X-ray structures from the protein databank and millions of SER data points from Campbell and other sources. In this respect, unlike most other computational approaches, ours is rather a virtual medicinal chemist than a virtual screener. Now, how does all this work? As shown on the left, here, the FRASE database is searched for protein environments that are similar to those in the protein of interest. When such environments are identified, the respective ligand fragments automatically populate the target protein. Finally, new ligands can be constructed by chemically linking those hit fragments. The technical implementation of the process is slightly different though. As you can see here in the center, the target protein is aligned with the FRASE database, which enables a straightforward comparison of every protein environment, as you can see here, with the respective environment in the target protein. Amino acids from the database and target environments are evaluated on both chemical and 3D similarity and 3D coordinate match. We were able to test the new concept in an ongoing project in the Center for Integrative Chemical Biology and Drug Discovery at UNC. The targets in this project are a small family of tyrosine receptor kinases, tyro3, AXL, and more, or just 10. Okay. In their normal lives, these proteins are expressed in monocytes to clear apoptotic material. However, in multiple cancers, they are hijacked to promote cell survival and constitute a sort of the cancer cell's immune system. This makes of TAM family members promising cancer targets. In fact, one more inhibitor resulting from this project is already in phase 1 clinical trial. The current goals of the TAM kinase project are to further explore their therapeutic potential. There is a sound rationale that compounds with varying intrafamily polypharmacologic profiles may produce varying therapeutic profiles. In particular, we recently received funding to investigate the potential of dual AXL-MER inhibitors. However, tuning the intra-TAM selectivity is an extremely challenging task. As you can see in picture on the left, there is only one residue position that shows intrafamily variation in the ATP binding pocket, and which is quite far from a typical TAM inhibitor as shown in the picture. That is why we decided to design inhibitors that would span both the adenine and backpockets as shown in the center. And the backpocket shows, in fact, a significantly higher rate of sequence variation that might be exploited to modulate both the intrafamily polypharmacology and improve the kinome-wide selectivity of new inhibitors. Our design strategy was to start with a small inhibitor binding to the adenine pocket and then use the FRASE database to identify fragments that would extend the compound toward the backpocket, like as shown here. So the first thing we did was to find a spacer pointing in the right direction. We identified several fragments featuring, so here are the fragments identified. So several fragments that feature alkyne group that seem to do the job. A compound featuring the spacer bringing an aromatic group to the entry of the backpocket showed promising results. Interestingly, this alkyne fragment was donated, so to speak, by AKT2, CHEK1, and MELK kinases that are very far from the TAM family on the phylogenetic kinase tree. Next, we exploited a set of phrases with aliphatic ligand fragments deep inside the backpocket, like these magenta colored structures in the figure. Embedding these fragments significantly improved the potency of the resulting compounds and showed varying intra-TAM polypharmacology. In particular, we obtained this AXL-specific compound here in the center. It's in fact the first AXL-specific inhibitor we managed to come up with since the beginning of the TAM project in the center. And again, the fragments were donated by a large number of very different kinases spread all over the phylogenetic kinase tree. A co-crystal structure was obtained for one of the novel inhibitors, thus confirming the structural rationale behind the FRASE-based design. Also, to assess the kinome-wide selectivity of new compounds, the UNC proteomics facility have run a proteomics assay, MIB-MS, where the selectivity is evaluated on virtually full kinome in live cells. Here UNC 1917 this one, a potent MER inhibitor, and adenine pocket binder was used as a reference. And it was found that UNC 4042, shown here, that is a novel pan-TAM inhibitor, is significantly more selective than UNC 1917, with only 27 enriched kinases against 62 for UNC 1917. And finally, UNC 4042 showed nanomolar potency in a broad panel of cellular assays. It was also tested in vivo and induced significant reduction of phospho-MER level in bone marrow leukemia mice model. So I show this slide again, just to remind that the current algorithm for the FRASE-based design for the FRASE database screening requires all the proteins in the database to be aligned with the target protein. Hence, fragments can be transferred and shared between members of a protein family with a similar fold, but not across families. And this seriously limits the impact of our approach, since promising novel targets may belong to smaller families for which no structure activity data exist yet. That is why our current work is focused on generalizing FRASE-based design so that the FRASEs and ligand fragments could be compared and shared across the whole proteome. To this end, we develop an integrated AI-based workflow, which will include 3 major components, which I'll briefly describe in the next few slides. The main challenge for the database screening is algorithmic. We need a fast technique to test whether each of hundreds of thousands phrases from the Proteome-wide FRASE database would match an unknown location in a protein of interest in one of count list orientation. The combinatorial challenge will be addressed through encoding of protein environments into a canonical string notation that is amenable to ultra-fast comparisons. This solution may lack specificity, but preliminary hit lists are further refined through explicit 2D and 3D comparisons of the database environments to those in the target protein. The main challenge for the next step is to quantitatively assess whether a ligand fragment belongs to a hit phrase, belonging to a hit phrase would fit in the target protein environment. And this fitting in may mean multiple things. For example, one question one may ask is whether the match between the structural environments detected by fast screening is correct. Again, whatever this might mean. Or another question is whether a ligand fragment from a hit phrase would be beneficial or harmful for the ability of a whole ligand it will be part of. So here we decided to assess the holistic usefulness of a fragment by using deep neural networks to learn interaction patterns of well-behaved fragments. And finally, the last step of our integrated lead generation platform. So generative methods are to be applied to produce valid 3D ligand structures within the target binding site. On this, we collaborate with Tropsha group to adapt his release approach to ligand generation based on 3D constraints. So just a quick recap to conclude. A new technology to assemble ligands from fragments directly in the protein binding site is being developed. Unlike most computational techniques that are virtual screeners, FRASE-based design is rather a virtual medicinal chemist. The new concept was used to design a series of in vivo TAM inhibitors with better intra-TAM and kinome-wide selectivity. Current developments are focused on FRASE-bot, a deep learning-based technology. And of course, our computational effort wouldn't have any value without practical applications in drug and probe discovery. So this particular project was a multidisciplinary collaboration involving many people from our center. And here all experimental work was coordinated by Xiaodong Wang. Other UNC groups include Shelley Earp Lab at the School of Medicine and the Proteomics Core Facility. And most of cellular assays in the in vivo study were performed by our colleagues from Emory, Doug Graham, Deb de Riker, and others.